Okay, all right. Okay, everyone. Just a very quick mic test to make sure that you can all see the screen. Great. So I see that the numbers are still climbing, so we're gonna give um the rest of the audience a couple of minutes before we start our talk for tonight. And I think I'm very glad to have Prof. Ruth with us tonight, who'll be sharing her expertise on obesity. Okay, so we will start in a couple of minutes. So let's give others few more minutes to just trickle in. Okay, everyone, we will start in 1 minute. Okay, all right. Good evening, everyone. Thank you very much for your time, for being here with us, and especially a warm welcome to Prof. Ruth for being here with us today. So I know that Prof. Ruth has a very packed schedule, so I'm really thankful that Prof. Ruth is able to provide some of her time to actually share to us some of the work that she is currently doing over at Copenhagen and Denmark. So first of all, I'm going to introduce Prof. Ruth first, who is very prolific. So Prof. Ruth serves as the Vice Executive Director of the Novo Nordisk Foundation Center for Basic Metabolic Research at the University of Copenhagen. So she holds a joint appointment as professor at the University of Copenhagen and also at the Icahn School of Medicine at Mount Sinai in New York. Her academic journey took her from a PhD at the University of Leuven in Belgium through postdoctoral training at the Pennington Biomedical Research Center and on to a research leadership role at the MRC Epidemiology Unit in Cambridge. And this reflects both her breadth and depth of her training and expertise. Prof. Lu has dedicated her career to understanding the genetic underpinnings of obesity and body weight regulation. As a founding member of the Genetic Investigation of Anthropometric Traits Consortium, the genetic investigation of anthropometric traits, she has led some of the largest gene discovery efforts for BMI and obesity undertaken to date. Her work goes beyond simply identifying genetic variants. She integrates genomic and proteomic data to define biologically meaningful obesity subtypes, with the ultimate goal of enabling more personalized approaches to prevention and treatment. She is also the principal investigation of Delphi, the Danish Precision Health Initiative, a remarkable 25-year longitudinal study following 10,000 individuals to build one of the most comprehensive multi-dimensional health datasets in the world. With over 600 publications and an H index of 179, Prof. Ruth stands among the most influential researchers in metabolic and genetic epidemiology globally. Please join me in welcoming Professor Ruth Lewis. Professor Ruth, over to you.
Thank you so much for the kind introduction, and thank you so much for having me. And it's a real pleasure to present today. Coincidentally, it is today the World Obesity Day. So it seems kind of appropriate to talk about obesity today. Should I swap screens? I don't know what you see.
I'm currently seeing the speaker notes.
Yeah, so I need to swap. Better? Did you? Oh, nothing happened.
Yeah, so I think that we are still currently in the speaker notes. Yeah,
Okay, you know what? I'm gonna unshare and maybe I need to
sure.
take this. Share this one. See, what about this?
Yeah.
Is that good?
Yeah.
Yeah. Okay, so my talk today is about using genetics in the context of personalizing obesity care. And it's actually only one part of the work I do. Typically, I also use genetics to learn more about the underlying biology. But today I will particularly focus on whether we can or cannot use genetics to really personalize obesity care. And I want to start with this story. This story is about a boy called Lucas Thompson. And I don't know the boy, but here in Denmark on Danish TV, they made a documentary about him a few years ago. And they titled the documentary The Boy with Eternal Hunger. Because indeed, pretty much from birth, Lucas had this unstoppable hunger. He could not stop eating, and his parents did not know what was happening. They talked to their doctor, they talked to the nurse, they talked to the school pedagogue to seek for help, but he kept gaining weight, and they were sure that something was wrong. And actually, in this picture, you see Lucas when he was maybe about age 3, and this is his older brother. So clearly, the parents knew that this was not what they expected based on their gene pool and environment. And indeed, by age 3, he weighed already 40 kilograms, which is about 3 times the weight that a normal toddler weighs. But that was also the age that genetic screening indeed identified the cause of his overweight and of his eternal hunger. Indeed, he had a mutation in the gene called the leptin receptor. And at that time, when they discovered that, there was not much they could do. It was just helping him to manage his food intake, but even though he felt hungry all the time. So her mom quit her job just to go pick him up at school, bring him to school, make sure that at night he would not wake up to open the cupboards and to eat. But even though the parents really took care of his food intake, he kept gaining weight. And by the age of 12, he weighed 120 kilograms. So what is the leptin receptor? So the leptin receptor is a protein that works in the leptin melanocortin pathway. So here you get a schematic overview, basically from the fat talking to the brain. This is the arcuate nucleus and this is the PVN. The fat releases leptin. Leptin binds to the leptin receptor in the arcuate nucleus, and this triggers a cascade of signaling effects. Eventually, the MC4R, the melanocortin 4 receptor, gets activated, and this informs the brain that the body has enough energy stored and you can't stop eating. So clearly in the case of Lucas, he has no functioning leptin receptor, so he never gets that signal. So he always thinks that he's hungry and that his body does not have enough energy. Interestingly, or luckily, while they were making the documentary, Lucas qualified to participate or to receive a drug called setmelanotide. And setmelanotide is a drug that acts, and it's not semaglutide, sort of the well-known sort of Ozempic or Mounjaro. This is setmelanotide. It acts directly on the MC4R receptor. It's a MC4R agonist, and in a way triggers the brain, make you think that you have eaten enough. And that's particularly useful if you have mutations in any of the genes that are precursors to MC4R. So, for example, in the case of Lucas, it would be effective because even though he has no leptin receptor, indirectly acting on MC4R, setmelanotide could sort of replace that signal easily. And indeed, he got setmelanotide, which resulted in kind of a nice weight loss during the time they were making the documentary. So over the following 18 months, he lost 20 kilograms, even though he grew 10 centimeter. And by now, Lucas, and this is of course a few years ago, he's a teenager, he's lost weight, he looks and feels better. He can now go on play dates, and more importantly, is like he actually is not thinking about food. It's like 24/7 anymore. So for me, this was actually a nice example showing that genetics, in this case, is in this case like, or I should say, is like the care of his obesity is nicely tailored towards his genetics. So because we knew that there was a genetic mutation, we knew the pathway, then it was in a way almost easy to tackle that pathway and then treat his obesity. But of course, the form of obesity that Lucas has is pretty rare. We call it a monogenic form of obesity, and the leptin receptor is one of the causes of monogenic forms of obesity. And there's a few more genes that cause monogenic obesity, such as mutations in MC4R, mutations in the leptin receptor, the POMCs. Many mutations within this pathway I just showed you cause monogenic forms of obesity. They are early onset, they're severe, and they contrast to the common obesity that we typically see among the population, and that is currently in a real like epidemic. But monogenic forms of obesity, they have a high genetic contribution. It's often a single mutation or a frameshift mutation in one gene. It has a big effect. It's pretty rare. It's highly penetrant. So if you are a carrier, or definitely like if you're homozygous, you're very likely to have this form of obesity, and it hardly does not matter what kind of environment you live in. Now, today, I particularly want to talk about common forms of obesity, but I thought starting with this story gives you an idea like how genetics indeed can be used in the context of precision medicine, because indeed for Lucas, the medicine he got was tailored towards the mutation he suffers from. But for polygenic obesity, where we have the genetic contribution is much more modest in the sense that the heritability is around 50%, hundreds, maybe even thousands of genes now, variations that act in many genes now contribute, not just one genetic mutation. And each of these variants has a small effect. They're very common. Many of us are carriers. The penetrance is low. There's no guarantee that if you carry any of these like low or small effect variants that you therefore have a higher weight. And the environment remains very critical. So today I really want to focus on this polygenic form of obesity, and can we use the genetics that we currently know of? Can we use that towards determining like who should get what kind of treatment, or maybe to better diagnose individuals with obesity? So I want to cover you a few aspects of precision health or precision medicine. So we know that there's not only precision treatment, often we think the right medication to the right person, but there's also precision diagnosis, precision prevention, precision prognosis, and precision prediction. So can we use genetic information to tailor the treatment, prevention, prognosis, prediction, and so on? So first, maybe let's talk about precision prevention and precision treatment. A little in the context of what we learned from Lucas, can we translate that to polygenic forms of obesity? Now, many companies actually do think that based on your genome, they can tell you what you should eat, what type of sports should you, or what type of exercise should do. For example, you may be familiar with 23andMe, one of the few companies that is very active in this field, but there are and there have been many other companies that claim that based on your genotype, they can tell you like what kind of diet and exercise you need. And here I'm just showing a few. I should say that many of these companies have come and gone or have been transformed to something else. But the claims they make are a bit bold. For example, this company says that if you are worried about losing weight, your genetic information will help you a lot. And they claim that we will be able to see your predisposition about which slimming diet is effective for you. The other company says, now you can eat a healthy diet that is best suited to your genetic makeup. Or another company that says, they provide a test that physicians can use to genetically match the diet and to make specific recommendations based on their genotype. So these are very bold claims, almost in the same trend of Lucas. If you have this genotype, you need this diet or you need this exercise. Now the question is like, how solid is the science behind this? And it's very often, if you go to these web pages, to these companies, it's very hard to get more information, like what are the papers that this science is based on, what are the genes that they use. So I decided many years ago, or not that many years ago, but several years ago, that I was going to participate and maybe get into it better. So I signed up for 23andMe. And I must say, like 23andMe is one of the few companies that is still around and that actually is doing a great work in like reporting their results. So here's my case study. 55-year-old woman, generally healthy, physically active, and wants advice for healthy aging. So I did this test, a direct-to-consumer test, DTC test, spat in a little tube, send it off, and 6 weeks later, I get my results. And I get my results on an app, very nice, informative app. So this is what the app looks like. If I then click on wellness, then you get more information. So it tells you about muscle composition and saturated fat index, sleep movement, and so on. So right now, I just want to focus on this, the saturated fat and weight, because here, if I click on that, then it tells me, Ruth, you are likely to weigh more on a diet high in saturated fat. And it gives you a little more information. For example, if my diet is low or below 22 grams per day in saturated fat, then my genotype does not really matter. Then my weight will be the same as the person with the other genotype. However, if my diet is high in saturated fat, meaning more than 22 grams a day, then because of my genotype, I will weigh 6% more than the other person who has the other genotype, even though this other person also has a diet high in saturated fat. And it's nice that 23andMe also provides these scientific details. So you click on the scientific details and you get the exact genetic variant that they study. And they tell me what my genotype is. I am a G homozygote. Now that's not super, it's in this gene called ApoA2. It tells you a little more. And then you can also see that it's actually a very common genetic variant. Like among at least European ancestry populations, 62% of the population carries this variant. It's a little less frequent in East Asian, but it's still very common in genetic terms. But what was interesting is that they also provide the papers on which they based the claims they made. So I looked up these two papers, and these are two very solid papers from colleagues at Rutgers University in Boston. And you see here nicely, this is data from the Framingham Offspring study, that indeed, as the saturated fat in diet is higher, then is the predicted BMI goes up, but it goes up in particular among this group of people. And this is the group of people that have my genotype. Whereas in people who have that other genotype, you see the slope is less pronounced. So the interaction between the genotype and the saturated fat intake is significant. It's just significant. But what is nice is that they actually nicely replicated that in a totally different study. You can again see that there is a nice interaction. But this is, of course, dangerous to interpret this in the context of making claims like if you have the way it's like 23andMe did it, because this is not data about weight gain. This is just observational data. It's like snapshot in time where they correlate results with each other. So the danger is here that, okay, we use saturated fat intake for this association. This is not response to saturated fat intake. So this might as well be replaced by maybe another factor that correlates with saturated fat intake. Maybe if I have a diet high in saturated fat, maybe I'm also less physically active. So maybe it's not really saturated fat intake that causes the BMI to be higher, but could be any proxy of unhealthy living. So just to emphasize that this data is not about weight gain, but just observational weight at one point in time. It's also just one variant. It's not, and this variant is very common. It's not like in Lucas's case, where it's a mutation that has a big effect and that disrupts the whole pathway in biology. This is just one little variant with a small effect. So what if I carry many other variants that are protective? So that's not taken into account here. And what do you tell people who do not have my genotype, but have this alternative genotype, at least for body mass index? You could almost say it's like, don't worry about saturated fat intake. With your genotype, you can eat anything. Maybe that's true for body mass index, but who knows, maybe that's not the case for, say, lipid levels or hypertension. So it's always a little dangerous to make a claim for one group, but not for the other. And then, of course, importantly, it's like even though the interaction is significant, that does not mean that therefore you can use this interaction to predict that this group of individuals will get a higher BMI compared to this group of individuals. So then I looked up, yeah, I should actually go back here. What's also remarkable is that maybe a little cheeky is that what they show here in this graph is not the actual BMI they observed, it's the predicted BMI. Yeah, so it's basically the residual line, and that might give you the false impression that is like a very clear-cut interaction. So therefore, I looked up a third paper of the same group, and they found the same interaction. This time, they did not use saturated fat intake, but they used dairy food, which could be a proxy for saturated fat intake. And now they show the predicted line, but they also the predicted BMI, but they also show the observed BMI. And now you actually see that, okay, the predicted line looks nice and there's a nice interaction as before. But now you also see that actually the scatter of dots of individuals is very much overlapping between the two genotype groups. So much so that it's going to be really hard to claim that this individual in gray would benefit from a diet in low saturated fat, whereas the individual next to this person would benefit from or would not be harmed from a high saturated fat. So this graph actually much more realistically gives you the idea of this interaction, and that it's actually not as clear cut as we might think based on the figures that we saw in the previous slide. So for if we want to use these kind of studies for these kind of interaction studies for prediction, saying like with your genotype, you should benefit from this diet, and with this other genotype, you would benefit from another genotype. Actually, we need interactions that are much more pronounced. So what we see in this previous study is we see like this interaction is statistically significant, but practically you see that there is still very much an overlap between the two genotype groups. So what we really need for prediction is that these two groups, these two genotype groups, are also very much separated, so that we with much more confidence can say that this group of people would benefit from a low saturated fat intake, whereas this group of people, it doesn't really matter. Anyway, this is just to say that even though some of these direct-to-consumer companies may make these claims that they can tell you what diet is good for you, what exercise is good for you, the science is really not there. So I don't really believe that the claims are really accurate. But as long as, of course, they don't do harm and they don't uh, they just give like healthy advice that is good for everyone, I think it's all right. But just to say, is like the science to do these kind of recommendations is still not there. What about precision prediction? Can we use genetics to predict who's at risk of obesity and who's not at risk of obesity? So what we do is we build genetic scores, and we build these scores on the number of genetic variants that have been identified to be associated with body mass index. And over the years, many genetic variants have been identified. It's like it started all with the one variant in 2007. That's the FTO variant. That's still the poster child of obesity genetics. But as over time, the sample sizes of our analyses increased, the number of variants that we identified has increased as well. And many of these large-scale studies have been led by the Giant Consortium. And you see, like I have not updated the slides since 2021, and it is actually because we have been working on a big meta-analysis. In fact, we now have the Giant Consortium itself has combined data of 2 million individuals. We collaborate with 23andMe, who share with us data of 3.5 million individuals, their customers. So now we have a meta-analysis of 5.6 million individuals, and I can tell you, it's like sometimes we have too much power. Now we have more than 9,000 variants that reach genome-wide significant association with body mass index. And it's been taking a long time for us to write up the paper to interpret really the biology behind it, because it's sometimes hard to define where a genetic locus ends and where the next one begins, because almost the whole genome lights up in terms of association with body mass index. So I'm not talking about the biology today, but well, I can talk about the use of all these genetic variants in a genetic score. So a genetic score, taking here an example, take these 7 individuals and take one variant of which we know that it's associated with body mass index. Take this individual, he is lucky in the sense that they did not inherit any risk allele from either parent. So you can have either no risk alleles, such as these 2 first individuals. You can inherit 1 risk allele from either your mom or your dad. You can inherit 2 risk alleles from both your parents, but that's the max. There's a 0, 1, or 2. And if you now do this for more variants, or you say like we do it for these 9,000 variants, or sometimes people take the whole genome, then you can add up how many BMI increasing variants a person carry. For example, this individual just over the whole genome scores 7, this individual scores 12, so this individual has a higher score compared to in particular this individual. So that's the way we can make scores, these genetic scores. It's slightly more complicated in the sense that some variants have a bigger effect than other variants, so we will weigh the score based on the effect size of each of these variants. So in that way, we can make genetic scores for any disease. So we did that also for body mass index. A score has most of the time a very nice normal distribution. Most of us have an average genetic susceptibility. A few of us are very susceptible to obesity. And on the other side, few of us are not genetically susceptible to obesity. So now we use these scores to predict who's at risk and who's not. But first I want to tell you is like how much these scores explain of the variation in body mass index. And this is an example from like quite a few years ago, but it was one of the first genetic studies where we showed an association between a genetic score and body mass index. And then you very nicely see that the higher, and yeah, first I want to point out that it's a normal distribution, but the higher the score, the higher the body mass index. And at that time, each additional risk variant was associated with an increase of your body weight by 440 grams. I'll come back to this graph later. But this is just to visualize how a score nicely correlates with body mass index. And now with the most recent genome-wide association data from 5.6 million individuals, we can make a new score. And now, and something we published a few months ago, last year actually, we are better than before at explaining variation in body mass index. So here I show the results of the previous score, which was based on 340,000 individuals. And in blue, the results of our most recent score based on 5 million individuals. And you see in these three studies, all of European ancestry, we explain now up to 18% of the variation in body mass index. And that's much more than what we did before. And this is pretty consistent across most ancestries. So we do pretty well with the same score. It's a cross-ancestry score in East Asian and South Asian populations, maybe a little less in like the general group of Asians where we did not have a good annotation from the Million Veteran Program. The Native Americans, it's less. And what is consistent is that, in particular for individuals of African ancestry, the score does not work well yet. And we see this not only in obesity, we see that also in other diseases. It's often because the sample size for African ancestry population is small, so that they contribute less and that they're therefore less represented in these scores. If we take the score and we rank all individuals going from the lowest score being in the 0th percentile up to the 100th percentile, then you see that individuals in this lowest percentile of the score, nobody has obesity. With the previous score, there were still 5% of individuals that had obesity, but now with our new score, nobody in this lowest score has obesity. In the highest score, at the 100th of the 99th percentile, 70% has obesity, much more than the general population. The general population, I think it's about 26% has obesity in the UK Biobank. Now, you can still wonder, like, okay, this is the highest genetic score. Oops, go back. Still 30% does not have obesity. It's like, despite the fact that they have the highest score. So that's another research question that we're trying to address. And see, like, why do some people with a high score have normal weight? We won't talk about that today, but it's another interesting topic. So can we use this genetic score now to predict who will have obesity in the future and who will not have obesity? So can we do a genetic test? So this means that a genetic test, the more risk alleles we have, the higher the risk of obesity. The fewer risk alleles we have, the lower the risk of obesity. So if we have a test that shows these two distributions separated from each other, then it basically means that the area under the receiver operator curve is 1, or the C statistic is 1. We perfectly predict who will have obesity, and we perfectly can predict who has not obesity. And that's the dream that we want, right? The worst score is that these two curves do not separate. And that's as good as flipping a coin, right? That's a score that really does not work well. And then the area under the receiver curve is 0.50. Reality will get somewhere in between. The question is, where in between are we? It's like, are we closer to 1 or are we closer to 0.50? So here are the results for obesity, class 1. That means like in people of European ancestry, it's above BMI above 25. I think people of East Asian ancestry is above 23. And here we see that in the previous score, we reached 0.65. Not so good, but at least better than flipping a coin. And now with the new score, we do a little better. The more extreme the obesity, the better the predictive ability, and particularly with an obesity class 3, it's like severe obesity, we are pretty good at predicting who is at risk of a future of severe obesity compared to who is not. Now the question is, if I now tell you what your score is, what are you going to do with it? At our age, it's like even if you're much younger than me, if you're in your 20s or even if you're in your 30s, you can still wonder, like, what do I do with this? If I tell you have a high score, should you panic? Or if I tell you have a low score, do you think you can just start eating? So that's always my question. It's like obesity is increasingly an early onset disease. So sometimes telling people in their adult life their high or low score may not be that informative. We sort of have reached an age where we have learned to live with our predisposition. So therefore, we start the questions like, at what age is this score really informative? And here you see, this is data from the ALSPAC study. It's a British study of about 7,000 individuals, and it's a birth cohort. So they're followed from age 0 up to 25 now. And here you nicely see that if you divide this group of individuals up at three levels, those in the lowest 10 percentile of the genetic score in red, in blue the ones in the highest level, and in yellow everything between, then you see that already by age 5, these three groups of individuals start to diverge. And it's that genetic susceptibility that is already present, it's like pushing through at that young age. Whereas before that, it does not seem to be that important. So then we ask, like, okay, when would this score now be informative? Say at age 8, you want to predict your BMI at age 18. Well, actually, at age 8, your actual BMI explains almost 50%, somewhere about 45% of your BMI by age 18. So your weight at age 18 is very informative for your weight at age 18. But the further away you get removed from from age 18, the less informative your weight is. Like by age 3, how much you weigh at age 3 does not explain much or cannot predict your weight at age 18. And that's just observed weight. That's like when you go stand on the scale. So what if we add a genetic score? So let's say it's like we add genetic score at age 8 on top of your observed weight at age 8. And you see that knowing your score at age 8, besides knowing your actual weight, does not add much at predicting your weight at age 18, right? But at these younger ages where your actual weight is not very predictive, maybe there the genetics is more important. And that's indeed what we see. Here we see that adding your genetic score to knowing your BMI actually significantly improves predicting your weight at age 18. So it's really at these younger ages where your weight is not yet informative of your adult weight that the genetics helps categorizing you like at more risk or lower risk. So this is really where genetics can help the most. And that's indeed important because, as I said, obesity is more and more an early onset disease. So the earlier we know people's genetic predisposition, the earlier we can start with prevention. And sometimes we think like, okay, if you have a high genetic score, that's it. You're set for a future of high body weight, but that's not fully true. It's like obesity, after all, is not 100% genetic. Obesity has a heritability of 50%, which means that even though genetics is important, the environment remains important. And this is actually what we showed nicely in that previous study in 2001, where even though you can like have a high genetic susceptibility, if you live an inactive life, you don't exercise a lot, then that association between the score and your body mass index is much steeper than if you live an active life. This basically tells you that even though you have a high genetic susceptibility, your physical activity levels can actually suppress that susceptibility by it's like 30 to 40%. So there is indeed still an effect of your lifestyle despite your genetic predisposition. Now, the next question is, can we use a genetic score in weight loss trials or in just weight loss, right? And so the first question here is like when we do weight loss trials or when we do weight loss using exercise and diet. The question is like, there's an average weight loss. Do people with a high score, if you think about it, would they be more resistant to weight loss, so they would lose less weight, or would they lose more weight? And after the trial, after they've lost weight, people gain weight again. Would they gain more weight or would they gain less weight? So that was the questions we set out to answer using the DPP, the Diabetes Prevention Program, and the Pounds Loss trial. So these are two kind of controlled trials that look at the effects of behavioral intervention. And what we see for both studies and combined, we see that people with a high genetic score, they lost more weight. It was actually counterintuitive to what I had expected. But at the same time, after the trial, they gain more weight. So it seems people with a high score, they seem to be more responsive to the environment. They lose more, they gain more. And maybe that is because when we do these genetic association studies, we do them in an adult life. And the BMI that we measure is actually not only a representation of a person's genetics, but also their response to a lifetime exposure to the environment. So this is sort of my reasoning, and that's why the score is indeed showing that response to this environment. It's different though when we talk about pharmacogenetics. If we think about the current weight loss trials or the weight loss interventions with medication, thinking about GLP-1 receptor agonists. This is a study that we also recently published. I think there's still a lot of movement in this field. It's like I'm citing this study to give you an idea, but trust me, I don't believe this is the last study, and I think much more new studies on this topic will come. But just to give you like an idea of the concept, here we asked the question, does the BMI score, and this was the older score, influence the weight loss in response to GLP-1 receptor agonists? And here we see that actually it does not matter. And in black, it's the meta-analysis of multiple studies. It basically is like if you have a high score or a low score, you will lose weight on a GLP-1 receptor agonist. We asked the same question for bariatric surgery, and there we show that people with a high score will lose less weight in bariatric surgery. We also tested the diabetes score, and actually we don't see much of an effect. Again, maybe a little bit in bariatric surgery, but not for GLP-1 receptor. We also look at separate variants, and these are all variants within some candidate genes, and actually I should add the genes, but I know this is PCSK2. I know there's some variants in GLP-1 receptor itself, but the bottom line is that in GLP-1 receptor agonist trials or bariatric surgery trials, we don't see an effect of a single variant. So just to say that a genetic variant or a genetic score, at least in response to medication, in response to GLP-1 receptor agonists, do not seem to determine the weight loss. But as I said, I think the field will be moving. I've seen press releases of companies claiming that they have a score that indeed affects the weight loss, but I haven't seen the studies that are behind the press release. So that's why I think it's still coming. So just a few last slides, and this is about using genetics in the context of diagnosis, precision diagnosis. In my view, is like precision diagnosis is actually at the core of precision health. Because if you can better diagnose an individual, then you probably also can better tailor the prevention and the treatment. Just a few slides on the idea or the concept behind it. So take these pink boxes are all genetic variants that are all associated with higher body weight. I know we have now 9,000, but this is just a snapshot of them. These same variants may be associated with waist to hip ratio, like a marker of fat distribution, but not all in the same way as BMI. So all these variants are associated with BMI, but this subset is associated with higher waist to hip ratio. This subset is not associated with higher waist to hip ratio, and this subset is associated with lower waist to hip ratio. And maybe you can add another phenotype, say like fasting glucose. And now you start to see like that there's another subset. So this subset are variants that are all associated with body mass index because there we started. They're also associated with waist to hip ratio, and they're also associated with higher fasting glucose. And then there's another subset that is associated with lower fasting glucose and so on. And you can add other traits. So you just start to see that there are clusters of variants, genetic variants. And with cluster 2, 3, 4, 5, 6. So what you can do is now you can calculate a genetic score for each cluster and then calculate the score in each of these individuals. And some individuals color more purple because they have a higher score for cluster 1. This is orange. Some individuals color more purple because they have a higher score for cluster 2 and so on. And maybe each cluster would benefit from a different kind of intervention or prevention. And this is just schematic, and this is just me sort of setting out the concept. Of course, this is still very much a concept. So in the last few slides, I want to show you an example where we did this type of analysis, but using genetics to subtype obesity in individuals with what we sometimes call the metabolically healthy obesity and individuals with the metabolically unhealthy obesity. Tell you how we did that. So we used data of UK Biobank, 552 individuals of European ancestry, and we created what we call the bi-traits. And what a bi-trait is we took 3 anthropometric traits, BMI, body fat percent, and waist hip ratio, and we took 8 cardiometabolic traits. And then we did the GWAS for all these analyses for BMI and body fat percentage, waist hip ratio, and for all these other traits. And then we aligned them 2 by 2. For example, we looked for genetic variants that are associated with higher BMI, but lower LDL levels. And we did the same for higher BMI and lower triglycerides, higher BMI and lower systolic blood pressure. So that goes against what we typically see, that there's typically a positive correlation between these traits. So we did that 24 times. So we had these 24 GWASs that identified genetic variants that are all associated with increased BMI, body fat percent and waist hip ratio, but lower levels of cardiometabolic health. Yeah, so we call it an uncoupling of obesity with its comorbidity. And we identified 205 loci in 266 independent signals. So now we can make a new genetic score, and this score is now for healthy obesity. Yeah, so it should be associated with higher BMI, but lower levels of blood pressure, glucose, and so on. And we can compare that with an unhealthy score, which is associated with higher BMI, but higher levels of glucose, insulin, and so on. So this score, these 2 scores would allow you to identify these 2 subsets of individuals already at an early age. And this is basically like confirming what we see. In red, we have this healthy obesity score, we call it uncoupling score, and in blue is the unhealthy obesity score of the body fat score. And then you indeed see that the score, the red score, is associated with, say, body mass index, fat-free mass index, body fat percentage, but it's not associated with, for example, liver fat fraction. If anything, it's associated with lower liver fat fraction and lower VAT over SAT. VAT over SAT is visceral fat over subcutaneous fat. So the fat distribution, even though it's associated with higher BMI, the fat distribution is optimal. Then this is in contrast to the blue line, where you see is like it's also associated with higher BMI, but it's also associated with higher waist hip ratio. There's actually no effect of VAT over SAT. What's important here is that the red score, even though it's associated with higher BMI, it is associated with lower levels of triglycerides, LDL, total cholesterol, HbA1c, glucose, systolic blood pressure, diastolic blood pressure, and also with higher HDL, which is good. Whereas the blue score is indeed the opposite. So this is just to confirm that These two scores, if you score high on them, one score will identify the unhealthy obesity individuals and the other score will identify the healthy obesity individuals. And then if we test the score using medical record data, then we nicely see that it's associated with the prevalence of obesity. But the red score, the healthy obesity score, is associated with lower levels of lipid metabolism, type 2 diabetes, hypertension, and so on. Whereas the blue score, sorry, is associated with higher levels of these diseases. Interestingly, though, if you now look at other diseases, the non-metabolic diseases, then actually you don't see a difference between the two score. Like think about emphysema, I think cellulitis, so all these weight-bearing diseases. There it doesn't matter whether it's the healthy or the unhealthy score. As soon as you have a higher BMI, you have a higher prevalence of these weight-bearing diseases. So the score, even though we call it healthy and unhealthy obesity score, it actually is only healthy when it comes to metabolic conditions. Yeah. So this is again a way of genetically identified people who have a different type of obesity. It's like to help subtype obesity, which again is at the core of precision health. So I'm going to summarize here. We have hundreds of variants, thousands of variants now associated with body mass index. The scores can be used to do the genetic predisposition. The polygenic score reflects a lifelong predisposition and is most informative in early childhood. We also saw the interaction with environment. So it's not because you have a high score that you are therefore destined to have obesity. And genetic scores can also help with identifying the subtypes of obesity, which is actually at the core of my research, but also of other people's research, using genetics for all kinds of complex diseases, of course, not only obesity. I didn't tackle this, it's a different topic, but even knowing your score and knowing like that you might be genetically susceptible does not mean that therefore it gets easier to change your behavior. I'll finish here. I'm grateful for a lot of collaborators, particularly also the Giant Consortium. We've been at the red line through my research and also many funders. And I'm happy to take questions.
Great. Thank you very much, Prof. Ruth. Really appreciate it. So, guys, thank you very much for your attention. So now it is the Q&A session. So what I'm going to do is I'm going to invite all of you to either put your questions down in the Q&A, or you can actually unmute yourself to actually converse with Prof. Ruth. So I already see that there's a couple of questions in the chat. So I'm going to read it out, and Prof. Ruth, perhaps you can actually respond to them. Okay. All right.
Yeah.
So okay, so the first question is by Robin, and Robin, if you want to unmute yourself to clarify, please feel free. Okay. So under standard metabolic conditions, what is the standard method to measure tissue-specific insulin sensitivity? What is the method and how reliably can they distinguish contributions from different tissues?
That's actually a very good question, and it's not totally in my field. What I do know as an epidemiologist, there are a lot of formulas and metrics that combine values from OGTTs, like going from 0 to 30 or from 0 to under 20, and like in all kinds of ways to indeed infer tissue-specific insulin sensitivity. And that's at an epidemiological level where we work with like a lot of data. I can imagine at a more experimental level, there might be other ways, maybe using clamps and so on. But it's indeed a very important question. It's part of the sort of the heterogeneity among type 2 diabetes. Not everyone has this like fat cell insulin resistance or liver insulin resistance. So yeah, it's a good question, but I'm not the full expert to really answer. But I know people in the field are working that as well.
Great. Thanks very much, Prof. Ruth. How accurate are genetic tests for what vitamins or mineral supplements are needed. So, for example, MTHFR gene mutation and B complex supplementation. Also, how accurate are these tests for insights on sleep, e.g., COMT variants?
Yeah, that is a good question. Actually, I used to use some of these examples in my teaching. Um, I I think sort of the MTHFR. Uh, some of these tests are actually pretty accurate because they are actually part of the biological pathway. Um, and it sort of, I'm very simply thinking of lactose intolerance, right? If you have like a variation in the LCT gene, then you basically, the dietary recommendation is avoid dairy. And that is, of course, because the variation is actually acting in a gene that is important for this um, for metabolizing dairy. So I can imagine that it's the same for these more um, molecular phenotypes. In the field I work, we talk about diet, and there, if you have a variation in a certain gene, that there's multiple ways to digest certain foods, right? Except of course of the dairy. So there, I think that's one of the reasons why the genetic tests that provide recommendations towards diet will never be very accurate. Um, um, because right because it's far more complex than just one molecule that is it's like part of this one single pathway. Not a great I didn't do a great job in answering it, but I think like when it comes to these more molecular phenotypes that where there's a variation in a gene that's part of the same pathway, I think the test could be fairly accurate.
Great. Thanks, Prof. Ruth. So I think the next one is by Matt Eckers Johnson. So are
Mhm.
there any good iPSC models for obesity currently?
Yeah, and that's a topic I didn't tackle today, but obesity biologically is actually a disease of the brain. The genes that we identify, the genetic variants, lie near genes that are expressed predominantly in the brain, probably influencing food intake, hunger, satiety, reward. And therefore, it's like we use like neurons, neuronal iPSC models. Some people use fat cells. Fat cells are actually more important, or adipocytes are more important when it comes to the link between obesity and its comorbidities. It's like, but indeed, the iPSC model for obesity is more are the neurons as a representation of the brain. Yeah.
Great. Thanks very much, Prof. Ru. So we have a couple more questions.
Mhm.
Again, just a reminder to everyone, please feel free to put in your questions in the chat, or please feel free to unmute yourself and you can actually verbalize and have a conversation with Prof. Ru as well. Okay. So the next question is by Dr. Chong Yoksin. So at what point, oh, sorry, I think someone unmuted yourself. Sorry. So I think Carrie, okay. So as I was saying by Dr. Chong, so at what point do you think precision health phenotyping becomes clinically actionable? Is there a minimum data depth threshold before personalized longevity recommendations are meaningful rather than speculative?
Mhm. That's a deep question as well. It's something we are trying to figure out ourselves. Um, Genetics is definitely only one component to precision health. Like as a geneticist, like 15 years ago, we were told like once the whole genome is complete, you have all the tools in hand to do precision medicine. And of course, we now know that that's definitely not the case. Like it's like obesity, diabetes, it's like all these complex diseases, mental health, um, neurodegenerative health. Like it's 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 complex, and like we talk about phenotyping, which is It's deep phenotype is indeed important. It's like, but it's only one part. There's a snapshot, right? So we have to really um, do the deep phenotyping over time. So it's hard to put a number or it's like quantify the minimum of depth that we need. But in my view, it's like it needs to be longitudinal and ideally, it's like the phenotypes should also be response phenotypes. I'm thinking in my field, response to a certain meal, for example. It's like not just fasting glucose, but maybe glucose in response to a mixed meal or an oral glucose tolerance test. So yeah, hard to answer, but it's a question that we also have, it's like as we are building our own deep phenotyping cohort.
Great. Thank you very much, Prof. Ruth. The next question is by Ang Keiling. So, would you know if there's any obesity gene variants that are associated to the gut microbiome?
Yeah, we haven't specifically looked at that, so it doesn't stand out. Um, Maybe there are some links, but I haven't really, yeah, I haven't seen any particular papers about it either.
Sure. Okay, thank you, Prof. Ruth. So, these two questions are from an anonymous attendee. So, could you comment on the mother's maternal health during pregnancy and the offspring's risk of type of obesity?
Yeah, the mother's maternal health during pregnancy definitely has an influence on the birth weight and the weight in the few weeks, months after. Um, but afterwards, the sort of the weight during and the weight gain during pregnancy does not necessarily influence the offspring's weight later on in life. The mom's weight before pregnancy is of course correlated because of a shared environment and the shared genetics, but actually the pregnancy weight gain seems to have more, it's like an acute effect on fetal growth and birth weight, but not later on. And that's the case for the boys, girls. Yeah.
Okay. So, yeah, so I mean, whoever posted this question, if you want further clarification, please feel free to unmute yourself or to add on and elaborate in the Q&A. Okay. Okay, so I think this one is another one. So given the heterogeneity within both PCOS and obesity, do we see genetic differences between lean and obese PCOS phenotypes? And could this overlap with the distinction between metabolically healthy and unhealthy obesity?
Yeah, that's a good question. I actually don't know the answer to that. It's definitely also an interesting topic, PCOS. Early, like yeah, I don't have the answer. I'm sorry, but it's definitely like very relevant. And it's like I've talked about the healthy, unhealthy subtyping, but there are even other ways of subtyping. So some individuals have early onset obesity and keep the weight on for a lifetime, whereas other people like it sort of um, slowly increases. There's different types of obesity when it comes to body composition, but also fat distribution. So indeed, it's like I mentioned healthy, unhealthy, but it's one type of subtyping. And I can imagine when it comes to PCOS, indeed, there might also be other subtypes that are genetically driven, not unlikely.
Okay, thanks very much. So again, just a reminder, guys. So if you want to further have a conversation with Prof. Ruth, please feel free to raise up your hand or you can just unmute yourself. Okay. So I think there's one more question in the Q&A box. Um, do you know of any current public databases that combines genomic data for polygenic risk score with longitudinal wearables and metabolic data?
Mhm. So UK Biobank does have now it's longitudinal, but I don't think the wearable data is longitudinal. But I think some of the metabolic data is longitudinal, and they are about to initiate more. They are about to recall individuals. So UK biobank, to some extent. Um, I'm thinking maybe the Human Phenotyping Project from the Weizmann Institute in Israel, but also not that long. Again, it's like wearables is kind of a relatively new thing, so it's hard to get it already to be longitudinal. Yeah, maybe my
Thanks.
knowledge does not stretch far enough here, but yeah, that's what I know. Yeah.
Thanks, Prof. So I think there's a request by this particular person. So Brian, again, Brian, if you want to unmute yourself, please feel free, okay? So Prof. Ruth, so this particular attendee has asked if you can go back to the slide with the pathway affecting MC4R. So the pathway MSH activates MC4R and AGRP deactivates MC4R. This seems like a homeostasis balance. Does the drug basically act in lieu of MSH? And is there adverse drug reaction?
Uh, this one? So the pathway MSH activates MC4R. Yeah. I don't, it does indeed bypasses and it directly activates MC4R. Adverse drugs. So it's not quite my field, but I do remember that the skin darkens. That's what I remember. And I think it was in some cases, sometimes the reason that people stop taking it. But I remember that being one of the side effects. The skin darkens. Yeah. Maybe there are more. It's now also a drug that's also used in individuals with Bardet-Biedl syndrome, and some of the other precursor POMC mutations. It's like, yeah, there's increasing numbers. So it's not a big market in a way because it's relatively rare, but it seemed to be pretty effective. If you give setmelanotide to individuals who do not have any of these mutations, it actually does not really work. So initially people thought like, oh, maybe it works in the general population, but no, it has a minimal effect on body weight. Maybe people lose over time. It's like maybe 3 kilograms, maybe over 6 months or so. So yeah.
Um, Thanks. So Brian, does that answer your question? Yep. Again, feel free to let us know and we can have a conversation with Prof. Ruth on this. Okay. Thanks, Prof. Ruth. So I think a couple more questions coming in. So this one is by Fares. In routine clinical practice, do you foresee a role for integrating polygenic risk scores into obesity risk screening in primary care, similar to cardiovascular risk calculators?
Yeah, and indeed, that's how I see it. It's a score that gives you an idea of your susceptibility, same as your actual BMI or your actual blood pressure gives you an idea of your risk at the moment, but also for your future risk. I think a genetic score could be a biomarker on your chart. The challenge is, of course, to introduce it to help general practitioners to interpret it, to help patients to interpret it. But I can see, and that's of course not only for obesity, but it's also for other diseases that we could have a score. The scores still need to get better. We need to still improve on the genetic scores. But the big advantage of a score is that it actually already can be calculated at birth or early on in life. So yeah, I think it should not be presented as like your destiny. It gives you a level of your susceptibility.
Okay. Thanks, Prof. Ruth. So I think another question. So how can ethnicity affect the PRS score? Can we just adopt the literature from one population for another population for obesity and generalize it?
Yeah. The score that we developed most recently is a trans ancestry score, and it should be usable across ancestries. And because of the sample size of the study, it worked pretty well in like Southeast Asian European ancestry. But despite the enormous sample size, it still does not work very well in African ancestry. So definitely ancestry is an important factor when develop scores. And so, even in our population, we try to make an African specific score. And even that score did still not work well, probably because the sample size of the African population subset of our big meta-analysis was too small to get really a good, powerful score. But indeed, ancestry is important when we work with polygenic scores.
Okay, thank you very much. Great. So that's all the questions from the Q&A. So any other burning questions that anyone would like to ask? Again, please feel free to unmute yourself, or you can put it in the Q&A box. Calling once. Anyone? Calling twice. Okay, all right. I don't think there's any other further questions from anyone. And I think once again, Prof. Ruth, thank you very much for your time, and also
My
to
pleasure.
everyone as well for your attention and time tonight with us as well. Thanks very much, everyone. Thank
Thank you for
you.
having me.
Thank
Take
you.
care. Have a nice evening.
Thank you. See you, Prof.
Bye.
Ruth. Bye.
Bye.